Okay, the endocrine system is a mechanism of the body's control system that's going to influence a number of different functions, mostly related to metabolic activities and of certain cells, most cells, and the means of hormone production or chemical messengers that can affect this process. The it's essentially a secondary system that's enhancing the function of the quick fix to any homeostatic imbalance, which would be the nervous system. It also is related to mechanisms that are ongoing, such as those found in the urinary system with regulation of fluid and electrolyte balance, for example. The endocrine system uses, as I just stated, these chemical signals or hormones, messengers, to travel throughout the body, target either specific locations or a multitude of different cells, and that targeting can occur in a number of different ways. This targeting often is going to involve certain types of hormones, and those hormones can be basic types that include those that are made up of the same material as a phospholipid bilayer, such as a ligand-based hormone or a steroid made of cholesterol. These two types, both lipid-based or lipid-soluble and steroid-based types, can move freely across the membrane and result in direct gene activation and the desired output from that particular cell. If the hormone that we're referring to is a peptide hormone or amino acid derived type, and is a protein, it's going to be dissolved in, or it will be soluble in water, and therefore, if it's water soluble, then it's going to have difficulty moving across a membrane that has phospholipid bilayer that is hydrophobic at its center. And therefore, a second messenger would be required in order to get activity in the cell or the genes within it to start cranking out a certain product or a desired mechanism. The hormone characteristics then that we will be referring to are going to be based on hormones that are produced in small quantities that affect either the site that it's produced in or some target location. It can be secreted in the interstitial space in the area that it's going to cause an effect in, or it can be transported throughout the circulatory system and act as a target, and a location will act as a specific target tissue with a receptor for its binding somewhere else in the body and cause the effect in that target location. The control mechanism for the endocrine system is associated with this picture, and here's the hypothalamus, the diencephalon structure that is has widespread control mechanisms involved in emotional response, fluid balance or dehydration, thirst control, hunger detection, sexual arousal, and all of these variety of different control mechanisms would be related and helping to control the endocrine system through neural cell bodies and in a couple of different ways. The first way would be that the hypothalamus itself could release from its nuclei, these two nuclei, the supraoptic nucleus here and the paraventricular nucleus, two neurohormones that are actually made here in the hypothalamus and are sent down through this hypophyseal hypothalamic tract and into the posterior pituitary, which is derived from nervous tissue. This pathway from these multipolar association neuron nuclei that we're referring to here, these multipolar association neurons, are going to produce in the supraoptic nucleus the ADH hormone, which is vasopressin, which is essentially going to target the collecting tubule walls and stop the loss of water when dehydration occurs. And as it does that, it makes those walls permeable to water that was being lost previous, and sends that water out into the interstitium, and it's picked up by the vasa recta, or the capillary bed that surrounds it. So reabsorption of water occurs, and as a result, with vasopressin or ADH release, you can also couple aldosterone function with that. That we'll talk about soon from the adrenal medulla or adrenal cortex, and affect blood pressure and blood volumes in that using that hormone. Above this is the paraventricular nucleus, which releases oxytocin, positive feedback related neurohormone that's going to target the uterine wall and the muscle that's found there to facilitate childbirth, and as it does that, it's going to cause uterine contraction and dispelling of the afterbirth, and then influx of milk for breastfeeding, followed by prolactin from the from the anterior pituitary. The third nucleus that we see here up in the that's vital to your understanding group is the ventral hypothalamus, which is under the control or which is going to control anterior pituitary function. So the two hormones that we just mentioned, ADH and oxytocin, released from these two nuclei, are going to travel down this tract, the hypophyseal hypothalamic tract, and move down into the posterior pituitary, where they would be picked up by this secondary portal system and sent throughout the body to their target sites. The ventral hypothalamus, on the other hand, is going to release releasing hormones here. Notice the axons are going to terminate here, and then release the hormones, either releasing or inhibiting hormones that will affect the underlying tissue, the anterior pituitary, which was in fact derived from the roof of the mouth and is an epithelial-based structure. The hormones that come from here are specific. There are 5 main hormones that are releasing hormones that are going to target release here of anterior pituitary hormones, and those would include CRH, it would also include PRH, GHRH, and GnRH, gonadotropic releasing hormone, to name a few here, and we'll have those in the next slide. So the cells that are targeted down below in the anterior pituitary will release 6 different hormones and target either tropically or non-tropically the locations of the body and cells of the body that we will discuss. This is the list of hormones that are released from that anterior pituitary gland, and if you notice, each of these hormones would be associated, as it says here in the picture, with the release of releasing hormones from the ventral hypothalamus to target their release. TSH is the first. It's going to be related to targeting in the thyroid gland tropically, and we'll look at that individually. ACTH targets the adrenal gland. The FSH and LH release is going to target gonadal function and proliferation of gametes and production of gametes there, as well as hormone levels rising. Growth hormone has a widespread effect that we'll spend some time looking at, and prolactin targets the mammary glands in mammals, and as well as another additional listing here, which would be the endorphins, which are natural pain receptor related hormones that are released from the brain. All right, so hormones of the anterior or the posterior pituitary, as I've mentioned, then a quick recap would include ADH, which regulates blood volume. Its secretion is also controlled by blood pressure changes as well, and dehydration mechanisms. The baroreceptors that are associated with this ADH release are specialized neurons found in the wall of the atria of the heart, large veins, carotid arteries, the aortic arch, and these all sense changes in blood pressure and help to detect this by a stretch reception or lack thereof. If blood pressure decreases a great amount, then ADH secretion is then stimulated, and we stop the loss of blood volume or volume in the filtrate and begin the reclaiming of that fluid into the bloodstream through reabsorption and aldosterone function. The antidiuretic hormone also is referred to as vasopressin, as I mentioned, and it's an anti-urination hormone, if you will, and that ADH is going to help to avoid dehydration, or at least help to reverse the effects of water overload or loss. Osmoreceptors, that are specialized neurons of the hypothalamus, help to monitor changes in the intercellular osmolality and monitor the solute concentration of the blood as a result. With high solutes, ADH secretion increases, and ADH stimulates kidney function to retain water and causes the change that resulted from ADH to allow for sodium movement and water movement that follows. With low solute volumes, ADH is not going to be released, thus causing water loss and the problems that resulted in its production in the first place. On the other hand, with the other neurohormone that's released from the hypothalamus, the posterior pituitary-related mechanism of release, oxytocin is also released. Oxytocin is a strong stimulant of uterine contraction. It regulates by positive feedback, so there's no direct regulatory mechanism shut off. And it's simply going to keep increasing in the bloodstream, which will lead to an increased intensity of the uterine contractions, which is necessary for childbirth and dispelling of the afterbirth following. It plays an important role in the expulsion of the fetus, as I've said, and during delivery by stimulating the uterine smooth muscle contraction, and then following all of this, it's going to result in the triggering of milk ejection in lactating women. Now, when you use the ventral hypothalamus, on the other hand, the ventral hypothalamus is going to be necessary for one of the major regulatory mechanisms that we associate with anterior pituitary hormone release, and that would be a hormonal mechanism. As we've said, the hypothalamus secretes releasing hormones that then stimulate the anterior pituitary to secrete hormones specific to either targeting of one location or of multitude of cells, depends on the hormone. In this picture, we see the thyroid gland, the adrenal cortex, and the gonads are all specific locations that would be targeted through the release of ventral hypothalamic hormones and would cause the anterior pituitary to target those individual sites tropically. The specific endocrine glands, we will go through each of these and the histology of those as we move along through this, but this method of regulation is referred to as hormonal regulation. The next regulatory mechanism is going to involve concentrations of certain things in the blood, of certain chemicals in the blood, and calcium would be an example of that. So as the calcium concentrations rise or fall throughout the bloodstream, you're going to see that there are certain cells throughout the body, the parathyroid chief cells, for example, that can detect this variation and fall in calcium levels, and as a result can result in can produce the parathyroid hormone PTH from them, thus stimulating osteoclast activity and as a result inhibiting osteoblast activity. The osteoclast would chew up the compact bone layer on the periosteal location and cause the calcium that hardens that bone to be released, causing blood levels to rise for calcium. This is a humoral response because both its turning on and shut-off mechanism would be related to blood levels and concentrations of that substance. There are a few of the different hormones that are going to affect the different sites in this way, and we'll talk about those further. The third type is neural control. So far we've had hormonal control from the ventral hypothalamus. We've had humoral control based on blood counts, or blood concentrations, such as calcium, and we have a direct neural stimulation like we see in the adrenal medulla with preganglionic neuron axons leading from the the lateral horn of the gray matter in the thoracolumbar region, T5 to T9, and heading out through the chain ganglia and innervating the adrenal gland with that preganglionic neuron axon that releases acetylcholine and then produces the catecholamine release that we've referred to or will refer to further with second messenger mechanisms, and that would include norepinephrine and epinephrine's sympathetic effects throughout the body. Notice that there is no postganglionic neurotransmitter release here. The release of epinephrine and norepinephrine is simply the product of acetylcholine from preganglionic neurons and neural control in that location. Now, the different second messenger mechanisms and the hormones that we see listed in this orange box here are going to be very specific. So if we have releasing hormones, catecholamines, or oxytocin and ADH, the neurohormones, then we're going to need to use, since those are all water soluble and have difficulty moving through this phospholipid hydrophobic portion of the phospholipids, the tails here, then we would have to, when these target, for example, TRH, when it targets the anterior pituitary to release TSH, as it binds, it causes this cascade of events if calcium is present, and that PIP mechanism that we see here. All right, so this mechanism is associated with each of these hormones. The catecholamines would be epinephrine and norepinephrine released from the adrenal medulla, and those would prolong the sympathetic response as well as help to inhibit or cause function during sympathetic activity in the different systems. So this is these are releasing hormones, and that we should associate with PIP mechanisms as well as neurohormones and catecholamine release. This box in this mechanism that we're looking at now is also a second messenger system. It's called the cyclic AMP second messenger, and it's going to trigger responses of target cells to activate enzymes, to stimulate cellular secretion, or open these channels. The hormones that are listed here are one, catecholamines that are associated with adrenal release. Two, they could be, most of them are anterior pituitary hormones that are tropic in nature. Or they could be also humorally related with concentration, like we see with glucagon and the regulation of glucose levels. But as well as PTH levels, also humoral. So on this list here, we're going to see all the same type of hormones, either peptide or amino acid, water-soluble type hormones that are going to need to target the surface of the cell and can't move freely across the phospholipid bilayer. So we'll come back and forth to this list as we go. Here's the image that you should associate with the anterior and posterior pituitary histology. These are the anterior pituitary cells here that relate to 6 hormones being released from stimulation by the ventral hypothalamic release of releasing hormones. So this is tissue that is derived from the roof of the mouth and is epithelial in nature. And then on this side we have the pituocytes that we see here, and the neurohypophysis that receives neurohormones from the 2 nuclei above, supraoptic and paraventricular, and those would migrate, move down that tract, the hypothalamic hypophyseal tract, and into this posterior pituitary area where the pituocytes here will aid in their uptake by the portal system that sends it throughout the body and targets a specific location. Here is another view of that same tissue, and that particular tissue is separated by a line down the center and is fed by the infundibulum, the pituitary stalk, one side being a neural tract pathway and the other side being derived from the roof of the mouth and consisting of epithelial cells. The first hormone that we're going to talk about, or loop that we should mention, is the thyroid hormone, T3, T4. T4 is thyroxine, T3 is triiodothyronine, and these two are related to the function of metabolic function in the body and an increase of uptake of glucose into the cells for metabolic increase. If we look at the histology and location, the location here obviously is in the superior mediastinum above the lungs, centrally or medially. It sits right below the thyroid cartilage in a male. The laryngeal prominence, the Adam's apple, is a characteristic feature, and as it sits there, it's sitting superficial to the trachea, the C-shaped rings of the trachea, and posterior to that, the esophagus. So it sits there, and on the back side of it we have the parathyroid glands that we'll look at in a moment that help with calcium balance. This hormone is helping with the regulation of 2 mechanisms. It's going to be, this gland is helping with regulating of 2 mechanisms, thyroid hormone and metabolic function, as well as its parafollicular cells helping to balance calcium levels in the bloodstream humorally. This is a picture of both the thyroid and parathyroid together. This is the tissue that we're going to focus on primarily, and as I magnify this, notice that the 2 structures, this is the large lumen of a thyroid gland and the ducts found within it and the colloid that's produced there, and here's the chief cell and oxyphil cell made up parathyroid gland next to it. And this would be a large vein that's adjacent, probably the jugular vein in the neck. As we look at this area right here and we magnify just the thyroid, you can see that there are cells that actually encircle that colloid filled area, the lumen of this structure, and this is the duct of this gland, and those cells are referred to, these cells here are referred to as follicular cells and are targeted by TSH. The cells that you see in between each of these circular ducts, such as here, or the cells right there in between the follicular cells, are called parafollicular C cells, and we'll discuss those with regard to the humoral regulation of calcium balance in the body. Okay, so now if we continue here and move on to the next slide, we see a closer view of these two glandular targets. This would be the thyroid gland that we're going to be talking about first, and here's the thyroid and parathyroid next to each other. We'll come back to this image. Alright, so this is a fully magnified view of the cells that we were just referring to that relate to the production of this colloid substance. Here is the follicular cells. So if you outline one of these cuboidal, simple cuboidal cells, it is the cell that would be targeted by TSH that was released from the anterior pituitary. It's targeted using cyclic AMP mechanisms, and the colloid we see in the lumen is in fact produced as a result of that second messenger system, and it is comprising essentially the essential components that will become thyroglobulin. As tyrosine adds iodine to it, it will then become the substance that we're actually triggering this loop for, T3 and T4, T4 being thyroxine and thyroid hormone, which controls your metabolic rate and uptake of glucose into your cells. Now if you look around, so that's the thyroid hormone production mechanism that we see here, and that's the colloid we associate with that mechanism. Now if you look right here in between these follicular cells, you can see cells that are sort of packed in between those follicular cells, and that's these cells here, 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 and here are all parafollicular C cells that relate to calcitonin release, humorally detected, and an increase in the production of bone from osteoblasts. Right, so you deposit bone when calcitonin is released because it starts out as a low level of calcium, a high level of calcium that triggers it. If calcium levels are too high with that regard to this mechanism, then calcitonin promotes osteoblast activity, it inhibits osteoclasts, and calcitonin would also cause the depositing of bone and then the lowering of blood calcium levels by depositing calcium in the form of hydroxyapatite into that bone and hardening it. On the other side of this mechanism is the chief cells you see here in the parathyroid, and those also are going to humorally, through the bloodstream, pick up a concentration that's too low in this case, and the low concentration of blood calcium would result in PTH release, targeting of osteoclasts, and those osteoclasts would then chew up the compact bone that we find on the outer periosteal layer that's hardened by calcium, and as it resorbs that bone using acids and enzymes from it, it's going to inhibit the osteoblast activity and cause a rise in blood calcium levels, which is essential to nerve impulse transmission, muscle contraction, and blood clotting, to name a few things. All right, this is where we find the parathyroid gland. So here's the backside view, a posterior view of the esophagus on the backside of the trachea. Here's the superior mediastinum, and that's where these structures are found. And here would be the back posterior portion of the thyroid and 4 specific parathyroid glands that sit in that location. So the parathyroid thyroid gland is often seen in the same slide, and its C cells of the thyroid as well as the chief cells in the parathyroid will help regulate calcium concentration. Those cells are seen a little bit better here. So if we go back to this image and magnify it, the cells that we're looking at here are chief cells and oxyphil cells. Oxyphil cells, the function of is unknown, but all of these abundant cells that we see here are what release PTH and help with 2 other mechanisms, vitamin D production and the kidneys balancing of that electrolyte to help regulate the mechanisms that we just mentioned. All right, so now as we move to this slide, this is the pancreas. Right? The pancreas is found, as you can see in this image right here, at the lower border of the epigastric region in the abdominal cavity. It's going to be hooking around or associated with the duodenum, the first part of the small intestine, and what's unique about this organ to all other, when you compare it to all others, is that it's both endocrine and exocrine. Its exocrine function is going to be associated with the release of digestive enzymes from the acinar cells that you see right here. And those acinar cells will be associated with ducts, the main pancreatic duct, for example, that you see in this image, and the release of enzymes through the hepatopancreatic ampulla into the small intestine for digestion and absorption of the nutrients from the food that we've eaten. It also is going to help to maintain a pH balance within that pathway of the gastrointestinal tract. Now, on the other hand, you can see right here the lighter area of this image, and if we magnify that, you can see the islet of Langerhans and the alpha and beta cells that comprise that structure. And this is the endocrine portion of this dual-natured gland or organ, and it's going to be controlling blood glucose levels with its alpha and beta cell release. Okay, so if we move to this image, you can see the mechanism of control, what it's related to, pancreatic release of insulin and glucagon. Insulin would be released and stimulate the uptake of high levels of glucose into a storage form called glycogen, and then storage of that glucose, bringing the levels down in the blood, in the liver and in muscle tissue. If levels decline and you have a low level of blood glucose, then glucagon would be stimulated, and you would call up those storage forms in the form of glycogen using a process of glycogenolysis to increase the levels if necessary. This is mainly a humoral mechanism, but is related to a number of different other hormones that we'll mention. Here's the islet of Langerhans magnified. This would be a nice view of a beta cell, much more abundant type of cell in this view, and the islet's beta cells would be responding to high levels of blood glucose and promoting the uptake and storage of those and decreasing levels in the blood. These cells, the 1, 2, 3 right here, are alpha cells, and those help to release glucagon in response to low levels of calcium and call up glucose or break down glycogen and increase glucose levels through either the process of gluconeogenesis or glycogenolysis. Now, here is the ovary. If we magnify the ovary, this is also going to be related to a control loop or a negative feedback loop that's related to anterior pituitary hormones, FSH and LH. This is a very nice view right here of a developing oocyte. And so this is the targeting location, the gonad in the female, and when you release FSH or LH from the anterior pituitary, you're releasing it to affect this location, and two things that go on here. This oocyte's development and the production of estrogen as a result of that, and the cells that are proliferating here, is under the control of FSH. On the other hand, when this egg during ovulation is released, you produce a structure that looks like this, and that's going to be the corpus luteum, a structure that both produces progesterone and serves to nourish the developing zygote if fertilization occurs. So FSH and LH are going to affect both hormone levels and follicular development or gametogenesis in this gonad. Additionally, in this male reproductive location, in the testes, which I guess I don't have that image. In the testes, you would find the same functions occurring, just slightly different. LH production in the seminiferous tubules would cause the Leydig cells there to produce testosterone, and FSH in that location would cause gametogenesis or sperm production, and both would be triggered by either a low sperm count, or low levels of testosterone in the bloodstream on a negative feedback loop. All right, this is a nice view of blood vessel. We'll come back to that one. Let's go ahead and go into the adrenal gland and wrap this up. In the layers of the adrenal gland, we find essentially 4 basic layers, and those layers are going to be seen over here at this in this area. The adrenal gland sits on top of the kidney. It's going to be a glandular structure, as the name implies, and it can release a variety of over 24 types of corticosteroids, as well as catecholamines from its inner medulla or medullary region. The classification of the hormones that we associate with this release would be mineralocorticoid from the outer zone, the zona glomerulosa. It produces the hormone that, 98% of the hormone that we're talking about here would be aldosterone, that we've mentioned already, secreted under low blood pressure conditions to increase the rate of sodium reabsorption by the kidneys and thereby increase sodium blood levels, as well as pull water with it to increase blood volume and blood pressure. The layer just beneath that, or the layer that we see right here, is the vertical arrangement of cells, and that's going to be the zona fasciculata. It's going to respond in production of steroid hormones by producing glucocorticoids, like cortisol. Cortisol is the major hormone from this region, and it increases fat and protein breakdown, as well as increases glucose synthesis through gluconeogenesis, and it helps with glucagon function from the islet of Langerhans and its alpha cells. It also will decrease the inflammatory response as healing occurs from damage to an area of the body. This is what the gland looks like. In many cases, you're going to see with a cross-section of this tissue 8 different layers that appear, but it's really just from outer to inner, a capsule of simple squamous epithelia, referred to as mesothelium. It's also called, if you're referring to the layers of the serous membrane, it would be referred to as the visceral peritoneum, found on the surface of this gland and covering multiple layers onto the kidney. The layers just beneath that capsule would be the 3 that we just mentioned. The zona glomerulosa is the outer layer, then the zona fasciculata, reticularis, all cortical regions, and then the innermost layer is the medulla, which releases catecholamines under neural regulation. This is what each of those layers look like. It's highly vascular, and the hormones produced in each of these layers are, like I said, all steroid hormones and can cause direct gene activation in the cells that they target in various areas of the body. The medulla is going to prolong the sympathetic response with its neural stimulation. So this is a nice picture showing the pre-ganglionic neuron axons that will innervate it and release acetylcholine to cause catecholamine release, like epinephrine and norepinephrine. All right, so moving on, this is the actual histology slide. Here's the zona glomerulosa here that releases aldosterone under the control mainly of renin release from the JG apparatus and stretch receptors in the kidneys filtering units. Okay, that's a mineralocorticoid. Just beneath that is the zona fasciculata here, and the zona fasciculata is what produces cortisol and helps with the functions that we just stated. It's a glucocorticoid, and it's often released under times of stress and related to other hormones as a result. This layer that we see as we move inward here, these bundles are going to be hormones that are producing hormone that is categorized either as glucocorticoid or gonadocorticoid, and the main gonadocorticoid would be androgens, of course, and estrogen that are going to enhance the effects of the gonadal produced steroid sex hormones. Further, we see the adrenal medulla, and the adrenal medulla, as I said, is the catecholamine producing area under neural control. Right, so if we look closer at this image, this is what I was referring to as I magnify this. Notice it appears as though there's 8 layers here, but it's really this is the outer capsule. Here's the glomerulosa layer that produces aldosterone. This would be the fasciculata layer that produces cortisol. And this layer here would be the zona reticularis that produces the sex hormones, or the gonadocorticoids. And all the way into the center right here would be the adrenal medulla and the catecholamine releasing area under control of sympathetic fibers, preganglionic fibers. And then as we move out to the other side, like I showed you in the picture, we would go in reverse order: reticularis, fasciculata, glomerulosa, and capsule. It's just appears as though it's 8. It's really just one side of that image that we saw here a moment ago. And so if you make a cut through here, you're going to get it. It's going to look like 8 layers when it's really only 4 doubled up. Okay, so if we magnify those further, this is what those areas look like close up. Here's the simple squamous of the mesothelial layer, the capsule of this gland. Right below it, you see these circular bundles. This is the zona glomerulosa close up that produces the mineralocorticoid aldosterone in response usually to renin release. And then below it is the zona fasciculata that responds to ventral hypothalamic release of corticotropic releasing hormone and ACTH production from the anterior pituitary that targets this area to release cortisol. Oh. Here you see the inner two layers. So this is the bottom of that fasciculata layer that we just looked at here. So if I move down further and magnify it, this is the zona reticularis that produces those gonadocorticoids, androgens and estrogens that are going to help with function, gonadal function, and sex hormone production throughout adolescence and puberty. Here, further in, we find that this group of cells right here are referred to as chromatin cells. The chromatin cells of the adrenal medulla are going to help with the production of catecholamines, epinephrine and norepinephrine from nervous innervation and neural regulation through preganglionic thoracolumbar outflow for sympathetic response. And then everything collects here in the blood and then is circulated throughout. Here we have a breakdown of all of the different hormone mechanisms that you have put into your table, which is on page 73 and 74 of your lab. And I hope that you put the time in and master each of these mechanisms and their interrelationships, especially with growth hormone and hormonal or humorally released and balanced endocrine function, such as what we see with the calcium balancing mechanism and glucose level concentration balancing. Study, and I'll talk to you soon.